Thank you very much, Nick, for agreeing to give a Build a Cell seminar, and you can get started.
Fantastic. I'm going to just share my screen, and I hope this will work without a hitch now. Now I've given, oh, sorry, I'm starting the wrong thing. Okay. Apologies for this. I just managed to press the Teams button, so hopefully this will disappear in a minute. But anyway, whilst my computer's sorting itself out. So thank you very much indeed for the opportunity to come and talk to you today. It's really, really exciting. It's been quite a obviously quite a difficult time to get out and talk to people and talk about the science that we love doing. So I'm going to give you a little bit of a sort of taste today of some of the experiments that we've done, sort of which are aimed, and it's very much a work in progress. Lots of the stuff that I'm going to be talking about is stuff that we're doing right at the moment. So obviously there will be bits that sort of I talk about and they're not finished, and there's no particular sort of conclusion to those bits, but I find them interesting. So I thought it would be nice to at least mention them. And what I'm going to be talking about is our sort of progress towards the idea of creating and using synthetic extremophiles, so particularly pressure tolerant membrane assemblies and some of the fundamental effects of pressure on model membranes. So just to give you an introduction to me, I work in the Department of Chemistry here at Imperial College in London. We're actually based on the new White City Innovation Campus. So it's a really exciting campus, which is about 3 years old now. We were one of the first departments to move out there. And it's a brand new area, which is growing extremely rapidly, which is set up with the ethos of co-locating academic innovation with our industrial partners and small-scale startups as well. So it's the whole campus there is full of academic research from blue skies through to really quite applied research, and that cuts across several different sectors. It cuts across the academic groups, it cuts across next door buildings, small scale startups, and the entire area has become a real sort of draw for sort of biotech. So Novartis have just moved across the road from us, and there's a huge development called Scale Space just down the road, which is for growing companies. My group sits within the membrane biophysics group, so we work very closely with a sort of consortium of 5 or 6 PIs, and we all co-direct the membrane biophysics group. We're all PIs who are interested in solving problems which are related to membranes and the use of biomembranes to create new materials. We've got a total sort of group size between the 5 of us of about, depending on the time of year, between about 45 and 50 people. So we're quite a large critical mass of people. I work as well within the Institute of Chemical Biology. In fact, I'm one of the co-directors of our doctoral training centre in the Institute of Chemical Biology, which is a cross-department organisation which brings together people from all different disciplines to work at the interface of physical sciences and biological sciences. So that's been a really, I think, a really fantastically successful and an exciting initiative, which has been a real focus for sort of activity in chemical biology and biophysics. My background actually is in soft materials. I started off as my PhD was in looking at formation of self-assembled catalysts and templated catalysts. So I've changed quite a bit since then, and I've been through quite a lot of different areas. But my interest really now is in structural changes in membranes and how we can both understand biology. I'll talk a little bit more about this in a minute. How we can understand biology, but also how we can exploit some of that fantastic innovation that's been brought about by evolution to create new materials. There's a real strong basis in my group of instrument development. So we love building new bits and pieces, and that spans from rapid prototyping. We've got a really absolutely brilliant advanced tax base just down the road from our new labs, which is a brilliant sort of place to sort of democratize innovation. So everybody at every level in the university and from outside the university actually can get involved there and build stuff. So we make loads of use of that, but we also do some more kind of traditional engineering and instrument development to build sort of more exacting pieces of equipment. And I'll talk a little bit about that when we talk about our pressure work today. And I'm also a huge synchrotron enthusiast. I make a lot of use, and my group makes a lot of use of particularly small angle X-ray diffraction at synchrotrons, both in the UK, Europe, and across the world. So that's a brief introduction to me and what I do and what I enjoy, which I think is obviously, to me anyway, one of the most important things. So, but what am I going to be talking about today? I'm not going to be talking about anything if this doesn't move on. Okay, so I'm going to be talking about our work in the sort of area of understanding extreme life. So how life can exist under extreme conditions, particularly conditions of pressure, and what that can tell us about how we can build new things and how we can exploit those adaptations to build new structures and use them. And these are just some examples of extreme life in the world. So this a water bear, which can be almost entirely dehydrated. It can survive the most utterly kind of unusual conditions for life to exist in. This is a salt lake, or super high salinity lake. And this, which I find absolutely remarkable, is some life down at the very bottom of the Mariana Trench. So this is nearly 11 kilometers underwater. There's pressure of over 1,000 times, nearly 1,100 times the pressure that we experience on Earth. And if you look at these things, they're little shrimps, basically. They are not unlike the sort of surface-like life that we have, but they can exist under these utterly crushing pressures. And so one of the things that we're really interested in is how they do that. So as I said, our sort of our work spans understanding biology, but also using sort of evolutionary inspiration. So can we use some of this understanding of how nature has adapted to extreme conditions to be able to develop new biotechnical approaches and extreme processing approaches as well? So I'll just touch on that, and we've got some what I think are really exciting active projects in that area right at the moment. So as I said before, our work, or the work in my group, really focuses around broadly in changing membrane structures. So looking at how biological membranes change shape, how they change organization within them. And that really comes down to sort of three pillars of what we're interested in: understanding biology, modeling structural changes, and inspiration for new soft materials. And like I said, we love building new bits of kit, and you'll see ample sort of pictures of the things that we've built scattered through here as well. And obviously, very happy to talk at length about any of those if they're interesting. So just one thing that I mentioned a moment ago, but it's worth appreciating, is that the pressures that the life has to put up with in our oceans are absolutely huge, and the range of pressures is enormous. So if you go down to the bottom of the Mariana Trench, so this is the deepest place known in the oceans. That's where James Cameron went in his little submarine a few years ago now. But there's actually relatively regular voyages and capture missions to capture organisms from right down there. And you see all the way down that there's life which despite the fact that it may look a little sort of terrifying at times, it's not unlike the sort of life that you see at surface pressure. And actually many of the membrane structural adaptations that take place membrane structural changes that take place to drive forward biological processes are exactly the same in these organisms as they are in surface life. So we're really interested in how this happens, how life can adapt to these sorts of conditions. And there's some evidence that there's kind of increases in the unsaturation in lipid chains, that there's extra kind of stabilization within the proteins in these, within the membrane proteins of these organisms, and also incorporation of other constituents into their membranes as well to help stabilize their structure and their function. What's really, really unclear is what the feedback mechanisms are that govern that. So there's plenty of evidence of what may, kind of lead to this adaptation from a compositional point of view, but how that composition is regulated is really, really unclear. And I should say we don't know, but what we're really doing at the moment is develop some tools and technologies to be able to try and address some of those questions. So I think I hope that this isn't too simplistic. It certainly wasn't obvious to me when I started in this area. But if you think about one of the critical building blocks of a membrane, the lipid phospholipids, what happens when you put them under pressure? And it's not absolutely completely straightforwardly obvious. If you've got a lipid like this and you put it under isotropic pressure, so you squash it equally from all sides, actually you don't get an isotropic response, and that's because it's an anisotropic molecule, and you've got these extended chains which the drive of an increasing pressure is always going to be a reduction in volume, and that is certainly at moderate pressures best achieved by squeezing together and ordering those hydrocarbon chains, and that may even be accompanied by a little bit of an extension of those chains, which is quite counterintuitive, that you get this, you squeeze something and it gets longer. But actually the overall volume change is still negative. So that's what's going on there. It's, like I said, it's because of the anisotropy of the arrangement of that molecule. And really, so what we've focused on for a little while now is looking at how this lipid structure change can filter through and build up to a change in the assembly structure of membranes, and how that might start to impact on some of their function. So one of the things, and like I said, I guess warned you at the start that there's going to be plenty of pictures of bits of instrumentation. So we built a system for doing high pressure, small and wide angle X-ray diffraction. This is effectively a big steel box where you can shine X-rays in one side and look at the diffracted X-rays coming out the other side. It can operate up to 7,000 bar. And quite importantly, for some of our sort of studies aimed at looking at the kinetics of structural rearrangement in membranes, you can jump the pressure anywhere in that range, 0 to 7,000 bar, in about 5 milliseconds, and then look at the structural evolution of the membrane after that. So this really is, it's a fantastic piece of engineering, which was headed up by a really talented mechanical engineer, Beatrice Gauthier, who worked in our group for quite some time. I should say this was a really, incredibly sort of fruitful collaboration with Beamline I-22 at Diamond Light Source. They were utterly fantastic development partners as part of this project. We've also, to try and address some of these questions, built a high pressure microscopy system. So in this, it's got actually very similar kind of specs to the X-ray diffraction system. We can go up to 4,000 bar. We can control it to within about 5 bar of pressure, and change temperature, and really, fantastically complements the Sachs wax system. And it means we can get a multi-scale view ranging from the sort of inter-chain spacing, which we get from wide-angle X-ray diffraction. We're looking at about 3 angstroms, right up to the assembly length scale. We can look at the lateral structure on giant vesicles. So looking at the tens, if not hundreds of microns regime, and we can cover that full range. So those were 2 of our kind of first bits of instrument development to try and address some of these questions in high pressure membrane biophysics. And I think one of the first things that's kind of quite important to sort of think about is what happens when these membrane lipids pack together. And apologies if this is kind of obvious, and I guess it is a little bit, but when you start to pack these membrane lipids together, you start to change the preferred curvature, or you reach a situation where you can use pressure to change the preferred curvature the monolayers which make up your membrane, and this is going to actually create different stresses. Once you pack two monolayers into a bilayer, you're going to be able to tune the internal stress of the membrane, and equally, that stress is something which is going to have to be either regulated or mitigated in deep sea and high pressure tolerant organisms. The other effect that you get when you start to pack these lipids together, if you look at slightly more moderate pressures and more moderate perturbations, rather than actually creating wholesale changes in the preferred curvature, you can actually just create changes in the lateral packing and the small changes in the monolayer and bilayer height as well. And that has some important implications a little bit later on when we start to look at some of this and the interaction with membrane proteins as well. So one thing that we were hugely pleased with ourselves about was that this does, it's measurable. It's definitely a measurable effect. You can see a compaction, lateral compaction of the membranes. And one of the things that we're actively looking at the moment is how the level of unsaturation within the lipid chains here can change the resistance to lateral compression under elevated pressure. And what we see actually in some really preliminary results, or what we think we see, is that either by changing the level of unsaturation in these chains, or in fact incorporating increased amounts of chain branching and branch chain alkanes into the middle of the bilayer, we can change the response, and in fact lessen the response of the lipid chain packing to increase pressure. One thing that I think was less expected was that you can actually create lateral structuring within a membrane. You can induce lateral structuring within a membrane by changing pressures as well. And that's not actually immediately obvious why that would happen. It certainly wasn't to us. That if you've got a membrane, a model membrane which consists of two different lipid types and some cholesterol, then you can actually cause phase separation of the higher melting point lipid and cholesterol by increasing pressure. And we think, and we're sort of reasonably confident, that's because the cholesterol sitting against the high melting point lipids actually adopts a smaller volume, or than the cholesterol being able to interact with all the different lipids in that bilayer. We can see this effect using small angle X-ray diffraction. So this is actually a low pressure X-ray diffraction pattern here, high pressure X-ray diffraction pattern, and you can see here peaks that correspond to two different layer spacings of our membrane, and that's shown sort of really quite clearly. And you can actually tune quite effectively and quite broadly the thickness of these two membrane patches, and we actually think that could be really useful in being able to create membranes which can change their structure and change their release properties as well under pressure. So that could be really important, for instance, in food processing, where actually high pressures are used in several different areas to process and preserve foods. It could also be extremely useful in areas of high pressure industrial processing as well. We can look at this as well as using X-ray diffraction. We can start to look at this using fluorescence microscopy. So what we've done here is to go to a whole different size regime. So this scale bar here is 30 microns. And here we're inducing lateral separation of two different lipid types and cholesterol by increasing pressure. The lipid that's fluorescently tagged here is the lower melting point, the more disordered lipid type, and so you can create really quite rapid changes in the membrane lateral phase separation as well. One thing that is, I think, really quite interesting is that these pictures down here were taken in, these pictures down here were taken in sort of at least pseudo equilibrium conditions. So although there's quite a lot of significant, kind of changes happening here over time, the overall structure of the membrane remains kind of relatively similar. And then if you take the pressure up, actually we're able to, as we saw in the previous slide, tune this thickness mismatch between the two different membrane types and drive increases in line tension and increases in the sort of extent of phase separation there, which again has lots of potential for things like concentrating proteins on the surface of the bilayer and being able to control the aggregation of materials on that bilayer as well. Just one last part on this, because I think it's a really, I hope you can see these videos, but these are some pressure jump experiments which are looking at these are real time. So these are a real-time video microscopy experiments where we do a jump in pressure, in this case from actually around 60 bar up to 200 bar to induce phase separation in these membranes. And we can jump down as well and look at the destruction of those patches. One thing that is utterly remarkable about this is that if you track the line edge or the extent of phase separation, I had a really fantastic PhD student who put together these routines based on kind of local thresholding, and she got this extremely robust method to be able to analyze each of these frames, which has taken it about, I think these ones are the full videos were taken at close to 1,000 frames a second. And she was able to track the progress of the phase separation, and it equates extremely closely and really remarkably to the X-ray diffraction pressure jump experiments that we were able to do as well. And that may not be, kind of massively surprising, I guess, at first sight, but the fact that these the changes in phase separation and the changes in structure that happen they match effectively across the angstrom through to micron length scale is pretty remarkable. And you can see the progress of these. This is actually the perimeter versus area fraction of these domains, and the area fraction as you go from 0 to 18 seconds through these experiments. One thing that actually, so this was I thought really quite a nice experiment. We were approached by two really fantastic collaborators, Sarah Keller and Sarah Veach, to try some experiments out using model anesthetics. And it's well known that pressure can actually change the anesthetic behavior of many anesthetics, and there were sort of quite a lot of experiments quite a long time ago, and there's real lots and lots of debate around what the root of this is, whether at first it was thought that it must be a membrane effect because there is such sort of diverse structural diversity in the anesthetics which cause this to this pressure reversal happens in, but there's also plenty of kind of evidence and thinking that it must be either a protein effect or in fact whole organism effect as well. But this was a really kind of a lovely piece of combined work where we were able to look at GVs, so giant vesicles, and cell membrane extract, so giant plasma membrane vesicles, basically cell blebs, and in both cases we're able to show that the extent or I want to get this right, that the temperature at which phase separation occurs is linearly related to the change in temperature, sorry, I should say, at which phase separation occurs is linearly related to the hydrostatic pressure that you apply. And so you can see in this plot here that you can, if you've got no model anesthetic, you get a significantly steeper gradient here than if you've got a model anesthetic there. And you can see here that actually, if you apply pressure, you get reinstatement of the liquid-liquid phase separation within both a giant plasma membrane vesicle and a giant unilamellar vesicle, where that had previously been destroyed here by introduction of a model anesthetic. Okay? And this hydrostatic pressure corresponds very closely to the sorts of pressures which are known in very old animal experiments to cause reversal of anaesthesis. Okay, so I just wanted to sort of move on. So that's kind of quite a brief run through some of the stuff that we've done on sort of structure of membranes and how that can be altered using pressure. And I just wanted to touch on some of the work we've done on some slightly more subtle effects. So we've done some work kind of quite recently, both in the pressure sphere and in cells actually as well, looking at membrane micromechanics. In this case, we can actually look at the thermal fluctuations that happen in membranes, and by using a bit of image analysis to track where these membranes are, look at the magnitude of those thermal fluctuations, we can extract the bending rigidity, so the energetic cost of bending these membranes. Because this only uses light, we can actually do that under pressure as well, and we can see really quite a remarkable increase in the bending rigidity, so the energy required to bend a membrane under elevated pressure. At the moment, I have a project student working on looking at the effect of the compositional changes which are thought to impact pressure adaptation and pressure tolerance on these sorts of changes. And the first sort of kind of slightly tantalizing results are that actually this change in bending rigidity as a function of pressure is reduced when you start to introduce things like the isoprenoids, which are implicated in adaptation of archaea, for instance. So actually that's, I'll skip over that. That's actually what I showed you earlier, but that correlates very, very closely. In fact, the thickening of the bilayer and the compaction of the bilayer correlates very closely with the effects that you'd expect that to have on the bending rigidity. So we went on after doing some of those experiments, think, okay, well, it would be great to be able to do some more optical measurements and be able to look at for instance, transport passive transport across membranes and how that is affected by pressure as well. So we've built this high pressure fluorescent spectroscopy cell, or it's actually a fluorescence and absorbent cell, so it's got two light paths through it. And basically you can shine light in through one fiber optic cable. We have a fiber coupled spectrometer which sits on the other side, and we can do fluorescent spectroscopy experiments. One of the things that I thought was kind of most interesting here, really coming out of these experiments, and these are relatively new results here, is that if we have a fluorescent dye inside here, which is pH sensitive, and then load up outside the vesicle. So these are small vesicles now in our fluorescent spectroscopy cell. We've got we can track the movement of protons across the bilayer here, and that is then we can look at the rate of change in fact, this is the rate of change of the rate constant, so the the rate constant, first order rate constant for the permeation of protons here, the rate of change of the rate constant with pressure. Okay, so it's slightly, so basically a higher figure here says that the membrane in terms of its proton transport, proton permeability is more changed with pressure. Okay. And what you can see here, actually, this is just a sequence going from ordinary kind of surface pressure type lipids through a range of different constituents which are thought to be important. Again, actually, this is in archaeal adaptation. And you can see that actually, as you start to introduce more and more of these constituents, you reduce the susceptibility of these membranes to change their properties, and particularly their proton permeation with pressure. So I think that was really quite an important thing. One thing that I thought was kind of really cool in terms of work that we've done quite recently, both from a technical perspective but also an interest perspective, was the idea that actually maybe you can start to use these changes in membrane thickness to be able to kind of structurally communicate, if you like, with proteins and peptides embedded within the membrane, and cause protein structure changes driven by membrane structure changes. One of the things that's quite important about proteins, they're actually relatively insensitive to pressure relative to membrane assemblies. So membranes are quite they're quite supple. They can be squashed quite easily. To create a reasonable, reasonably large or reasonably detectable change in a protein, you need to be up at least around 1,000 bar. And you can see what we've seen earlier is that even with kind of 50 bar, 50 times atmospheric pressure, you can create really quite clearly observable effects in membrane. So there's a real disconnect between the intrinsic susceptibility of membranes and proteins to pressure. So we were interested in whether you could do something like this and whether that was something that would be measurable. And actually, we tried that in collaboration with the CD beamline, the circular dichroism beamline at Diamond Light Source, and we're working with orientated CD spectroscopy. So that's basically sort of based on the idea that if you have a helix, protein alpha helix, which is orientated with or perpendicular to the incoming UV radiation for a CD experiment, you will get a different signal out. So we made up a whole series of orientated bilayers, loaded them into a CD pressure cell, and loaded in a model protein. The protein we used was a protein called MSCL, mechanosensitive channel of large conductance, which we have used pretty extensively in the past. Got quite a lot of experience with using it, and you can see here, or actually probably more clearly here, that there is a really pretty significant change in the tilt angle of the alpha helices that make up the bundle, the pore bundle of MSCL, and that correlates really, excuse me, really quite nicely with the lattice parameter or kind of membrane thickness of the membrane. What we're doing at the moment is to do some experiments, and literally these are taking place at the moment, to look at a calcine efflux assay. So look at the release of a fluorescent dye, self-quench fluorescent dye from these types of membrane protein assemblies, and to be able to measure the release of material driven by the changes in the protein assembly. So I think really what we've been looking at here is a high pressure change that's caused the membrane, and then that structural communication with the membrane effectively acting as a structural amplifier to generate protein structure adaptation, and clearly then those sorts of changes in membrane thickness are going to be important in mediating or mitigating the effects of pressure in biology as well. Just one thing that I wanted to mention, this is my last slide, is that we're actually using or starting in a brand new project, which has just been funded, to start to use some of these sorts of concepts along with work that we've done in the past and in collaboration with Yuvali Lani and Oscar Says here at Imperial as well, looking at building up multi-compartment assemblies and building up sort of multi-vesicle assemblies, and being able to try and engineer some communication, some pressure-driven communication between compartments as well. So that's work which is planned and not yet started. So I'll look forward to sort of saying more about that when we've got something to say. So hopefully in this sort of brief run through, I've been able to just kind of touch on some of the things that I think are really, really exciting, really interesting in the relatively simple effect that pressure has on membrane building blocks, and how that can feed through in some pretty sort of important and in some cases relatively complicated effects on the assembly. And I think I'm not sure if we're getting any closer to understanding what the feedback mechanisms are, but we've certainly got some potential candidates for what those feedback mechanisms might involve now. So clearly, this is not just me sat in a lab. It's all sorts of people who have been absolutely instrumental in this work happening. There's my fantastic collaborators at Imperial, the people at Diamond Light Source, at Beamline I-22 particularly. Philip Auger's group at INSERT in Lyon, Roland Winter and Christoph Jarak at University of Dortmund, who have been utterly brilliant long-term collaborators and hugely kind of inspirational in the high pressure field. And like I said, Sarah Keller and Sarah Beach at the University of Michigan and the University of Washington, who were really brilliant collaborators working on the anesthetic work as well. Finally, thank you very much indeed for listening, and I'll be very happy to take any questions that anyone has.
Thank you very much, Nick. I have a few questions myself, but there's a lot of questions in chat. Can you see chat?
I think I can. I've just stopped showing my screen so I can see.
If you could read a question before you answer it, that's going to work for people watching the recording. Definitely.
So let me start with I'll start with the first one. So the chain packing results are awesome. That's very kind indeed. Thank you. Does this scale with what's observable in natural extremophile membranes? So one of the issues actually with natural extremophile membranes is getting sufficient material to be able to do some of these X-ray diffraction experiments on. So we need kind of 20 milligrams of material or so, and we have been able to do that in Archaea, working with a group in Lyon who can grow them. And we've actually just started working with a group in China as well, who have strains that we're setting up to grow in the lab as well. And certainly from the archaeal membranes, yes, they do. And the compaction of the lipid membranes is, I guess, in some ways predictably, reduced when you start to do things like increase chain branching and increase the concentration of isoprenoid compounds within the membrane. It basically just kind of it doesn't rigidify the membrane. It actually makes it less rigid to bending, but it makes it more resistant to lateral compression. Well, I hope that helps. So we've got another question: how much external pressure translates to increase in pressure inside a cell? How much membrane protects the cytoplasm? So that's a really, really good question, and not very much at all. So you can effectively assume that any pressure which you apply outside is transmitted to the inside, and we know this from some of our microscopy experiments, where we've looked at the just basic radius measurements actually, and you can see that there is effectively kind of immediate equilibration. So actually the sorts of molecules are amazingly resistant to pressure, really, relative to whole cells. And so one of the important things though is that the reason that you don't get just sort of whole scale squashing of the cell is that the water inside them, or the aqueous phase inside them is really pretty. It's got quite a high bulk modulus, so you don't get huge changes in volume, but you do if you increase pressure to 7,000 bar, you get about 30% reduction in volume of water, so it can be quite significant. So we've got another one here. Do you know if organisms living under pressure have any changes to cytoplasm viscosity? That's a really, really good question. And we have a collaborator, Marina Krimova, who I've worked with for a long time on actually trying to implement some fluorescence lifetime imaging experiments using high pressure. There's some real technical challenges with that, with being able to get high NA objectives, being able to get basically a confocal microscope compatible with the pressure cell. We are starting to get there, but she has these fluorescence rotor probes, and as soon as that technical sort of barrier is overcome, we'll be able to answer that, I hope. I suspect that it would increase cytoplasmic viscosity, at least a little. Will the effect of pressure on a cell's outer membrane differ from that of the membrane-bound organelles? That's, again, really a really good question. And in all honesty, I don't know. I suspect that the sort of adaptations, at least the property-based adaptations, even if the compositional adaptations are slightly different, would be similar to maintain the sort of transport properties and the basic fundamental integrity as well. One thing that maybe I didn't mention, and again, sorry if this is obvious to anyone, but if you and I, with our lack of adaptation mechanisms, went down to the bottom of the Mariana Trench, many of our membranes would gel, and we would kind of sit down there like a kind of block of butter without any biochemistry going on at all, really. We would lose our fundamental kind of structural integrity. So, so that is something which would have to happen across all different membrane types. But I don't know, and kind of one of the reasons that we don't know is that there's a kind of another step up in terms of the difficulties with extracting sufficient material when you start to look at organelles compared to the full cell. So do high pressure adapted membranes lose stability or permeability at 1 atmosphere? So again, a really, really good question. And from what we've seen from archaeal membranes, no, they don't actually. And from a kind of a model membrane biophysics point of view, that is related to the fact that they seem to the archaea, which we've been working with, seem to increase their proportion of isoprenoids, and they are fairly well known to hugely broaden the fluid phase regions in model membranes. So in those situations, they don't. There absolutely are some organisms which are obligate barophiles, which have to, which can't survive at atmospheric pressure. There are many that are able to change their tolerance when they're grown at different pressures as well. So I have to say I don't know about the fundamental membrane properties, but there certainly are organisms which do not survive being brought up to the surface. Would I be able to speculate what is the pressure limit for current life building blocks, high pressure but low pressure too? Another really excellent question. Actually, one of the things that we're working on in this new project I've been kind of recently awarded is to look at the extent of adaptation that possibly could happen on a compositional kind of or from a compositional point of view, with the idea of being able to look at the potential for life to survive down in kind of the deep rock environments as well, which undoubtedly it does. But there obviously are real questions about how far that could they could survive in terms of pressures, which can be significantly larger than the pressures at the bottom of the ocean. And we're looking at that at the moment. I suspect that there is at least potential for doubling, if not further, of the pressure limits that potentially things could adapt to, just based on the kind of membrane biophysics of the building blocks. In terms of low pressure is not fundamentally, from a pressure point of view, too much of an issue. What is an issue is dehydration, based on the evaporation of water caused by low pressures. So that's a slightly difficult question, but from a fundamental kind of pressure point of view, low pressure's not so much of an issue, because you've only got one bar to go, and it's not so different. You're reducing the pressure down there. I hope that sort of kind of answers the question. Can you comment on the effect that pressure has on membrane permeability? Yeah, so we've got some actually, like I said, we've got some new results literally just last week on this as well, and there is a fairly clear trend where, as you increase pressure on a DOPC membrane, so a absolutely kind of completely standard US type membrane, you reduce the permeability to protons. So you pack the lipid chains more tightly, and that reduces the proton permeability, and that's quite clear from the results that we've been getting, both previously, but now with these sort of even better results, which are a really talented project students been getting over the last week or so. And so, yeah, there's a reduction in membrane permeability. If, like I said before, if you start to incorporate more isoprenoids, if you start to incorporate more chain branching in general, then you seem to reduce that increase in, you reduce the reduction in proton permeability. One thing that we don't see at the pressures that we're looking at is fundamental disintegration of the membrane. So we control against that with a or we take a control measurement of self-quenched dye release from these membranes as well. So building on previous question, do people look at extremophile adaptations in other ways too, at low pressure, something like bacteria in the upper atmosphere? So there is lots of work done on a whole range of extremophile adaptations, and in terms of kind of looking at those sorts of environments, probably a more fundamental issue for those types of organisms is lower temperatures and reduced water as well, so dehydration. So yes, absolutely, people do. They do, but in terms of our sort of technology, it's not. I think the pressure would be less of an issue, less of a fundamental issue for those organisms. Okay. Over to the last two questions here. Would membranes with higher protein content and sugar structures be more resistant to high pressure? Could ultimate high pressure extremophiles have some sort of protein-based membrane, like some people speculate, for origin of life stages? So that's a really, really interesting question. I should say I don't know. I know that there are quite a lot of membrane structural changes which take place when you introduce large amounts of sugar. There are also some critical sort of compounds which tend to be found in high pressure organisms, which are thought to stabilize their proteins as well. That's when you get to really very, very high pressures. and they effectively act like antifreezes, so to maintain protein protein flexibility. So I think it's absolutely something that is possible. I think, you know, certainly in the work that we've done, the membrane structure is sufficient to be able to you know, kind of maintain at least structural integrity, and that's not just holding together, it's also being fluid enough to support some dynamic biostructural changes as well. The kind of standard membrane structures that we're all used to are sufficient to do that. But absolutely, you know, there are lots of other ways to sort of compartmentalize things, and it's entirely possible that some of those might be even better. I have a practical question. How do you grow high pressure extremophiles? Are they obligatory high pressure? How do you grow them in pressure reactors all the time? Okay, so that's a really very good question. The work that we have done with the Philip O'Shea's group in Lyon, and the work that we're planning to do is using non-obligate barophiles, so they will survive for a little while at atmospheric pressure. They won't thrive, but they will survive. And so what you do is to load them up into a piece of heat shrink tubing, effectively, with a CO2 independent and pressure independent media, and you put them under elevated pressure and you leave them there. It's quite kind of low tech. It's quite obviously you need things that are going to be able to cope with those sorts of conditions. You can't go in and exchange the media. You can't control the dissolved gas levels or the pH. So you need things that are going to work reasonably well in there. But then actually quite, you know, there has been some reasonable success in the two groups that I mentioned, but also with, for instance, Doug Bartlett's group in Canada, growing these sorts of organisms as well. And that is the end of the chat questions. Thank you, everyone. That's really stimulating.
Thank you so much. I have a question that's kind of related. I guess you partially answered that when someone asked, does the pressure translate into the internal pressure? So the metabolism is basically, obviously to some extent, pressure insensitive or pressure agnostic, because the pressure doesn't translate, right? As long as you protect your membrane, if you're an extremophile, as long as your membrane protects you, the metabolism inside goes as normal.
Yeah, absolutely. And I think one of the things that's really important here is that the reason that we are targeting the, you know, sort of membrane changes is that membranes are the softest structures, really. You know, they have these, you know, kind of, they have quite a lot of it's not spare space even, it's spare conformational freedom in those chains, which can lead to quite large changes in structure based on relatively small changes in volume. So that's so, but kind of many other molecules in general are, like you say, they're pretty pressure agnostic, because there's not huge volume changes when their reactions happen and when their structural changes happen.
Yeah, that sounds really cool. All right, thank you so much. If there are no more questions, I want to say thank you again, Nick.
Well, thank you very much indeed. It's absolutely wonderful. Thank you. Okay, great.
And thanks everyone. Bye.
Bye-bye.